watch the pupils throat towards the end. Okay, so I just should say as an aside, since Sam mentioned my interest in music, which is very deep, that it's kind of one of the cruel jokes of nature that in birdsong people end up working with zebra finches, which have this. It's hard to regard it as a musical song. The females do, but sort of it has rhythm and little else. But the neat thing about this is you can see from the pupil's behavior is that he's peering. He's behaving as he's trying to attend to what's going on. He doesn't really know what the state is. But at the very end, he also does this little sub-vocalization. And that's also, I didn't point that out in the first video, but that's not uncommon, that the pupil will engage in some kind of vocal motor activity that doesn't produce sound around the time the tutor sings, but usually immediately after. This technique disrupts activity in the pupil's brain just when the tutor is singing. And you can then take, in the same design as I just described for the NMDA receptor blocking experiments, you can take an isolate bird, express channel rhodopsin, and then use tutor contingent activation of channel rhodopsin in pupil's HPC for a limited time during this phase here, and then remove the tutor, raise the birds up to adulthood, and the channel rhodopsin treated birds again learn nothing at all. They behave as if they were isolates, and the control birds learn quite well. In fact, some of the controls involve using the tutor singing to drive optogenetic activity in the pupil's HPC after the tutor's been removed. So they get the same pattern and amount of stimulation in HPC, but it's just not concurrent with the presence of the tutor. And again, the sonograms show really disrupted copying. So here's a control that got channel rhodopsin activity, but not while the tutor was present. It makes a very good copy of the tutor song, but these birds where channel rhodopsin was activated while the tutor was singing don't copy at all. And so these experiments limit in space and time HPC's involvement in encoding this experience. Okay, so as I mentioned, the temporal resolution is good, but it's not great, and we were curious, based on what's known about the premotor role of HPC in generating a precise timing signal, whether or not we could further elaborate on these experiments and determine whether or not HPC also plays a role in precisely encoding this experience of the tutor song. And for that, we turned to an electrical stimulation method where we get very, very temporally restricted stimulation in pupils HPC. Same experimental design as I mentioned. And when we targeted just a single syllable in the tutor song and raised the bird up to adulthood, he failed to copy the targeted syllable, but he copied the flanking syllables perfectly well. So this says whatever HPC is doing in this encoding process, whether it's a site of memory storage or somehow coordinating with other parts of the brain to encode this memory, it's doing so in a very, very precise manner. So one attractive idea is maybe, when the tutor's singing, this little clock in HPC is running and helping lay down a calibration signal for this memory, or maybe the auditory experience is being directly annealed to this timing signal in the pupil's brain. And I again make the analogy to music. I switched from being totally sheet trained, by that I mean, reading music trained, musician, as a classical musician, to trying to play more and more by ear as I've gotten older. In fact, now, try to do that all the time. And I found it interesting. One of the ways to learn if you're trying to just copy what somebody else is playing is to actually forget about what the melody is like, what the spectral stuff is like, and just get the timing information right. On a guitar, if you start to get the timing right, then a lot of the other stuff starts to fall in place. You kind of find where to hang the notes, is the way I think about it. So, maybe this is information about how the pupil's brain is engaged at this level. Maybe, this part of this timing circuit directly interacts with the auditory stream to create a memory. Okay, and this is just some of the behavior again to show disruption of the targeted syllable and good copying of the flanking syllables. All right, so any questions? Yeah, Ron.
So what's the control for just hearing? In other words, does the bird actually hear what the tutor is singing when the stimuli are being provided, whether they're the optogenetic one or whether they're the electrical one?
Yeah, I mean, we think at this level of the brain, we block here.
You think you get blocked?
Yeah, we think at the level of HVC. So we haven't done recordings in lower levels of the auditory system like the auditory thalamus. My guess is that auditory responses are intact at that level, but you're getting at a very, very important point, which is how does this part of the brain interact with the auditory system? And the punchline is it does in a very interesting way. We don't know the details of it yet. It's long been assumed, and a challenge for songbird researchers is that it's long been assumed that this circuitry is organized in a feed-forward manner. There's always discovery represented as a feed-forward circuit. And the auditory stream goes in, the motor stream comes out, but in fact, there's both very heavy recurrent architecture in the motor components of the circuit, just like you find in the mammalian brain. There's also recurrent entry into the auditory system from HVC. So when I said that HVC may be functioning as a calibration signal for the auditory system, I was alluding to is the possibility that HVC transmits a signal back into the auditory system. And we know that there's an anatomical locus for this. And that somehow is working with an incoming auditory stream to encode the memory. We don't know how deeply that signal penetrates into ascending parts of the circuit. My guess is not very far. We think it's at a high level of the auditory system that this interaction occurs. But that's definitely a possibility. But, and I should add, the NMDA receptor blocking experiments point more specifically to HVC being at least part of the network for encoding the memory. We, at least as far as we can tell, electrophysiologically blocking NMDA receptors doesn't change aspects of the spontaneous firing or song-evoke firing in HVC. So, at least in anesthetized birds. So, we think that says that maybe the spine enlargement we're seeing is really NMDA receptor dependent mechanism. It's happening in HVC. It's encoding as part of the representation there. But that it could be more distributed or downstream of HVC is definitely a possibility. All right. Other questions? I just have a few more things to talk about feedback. So, just, the last section of the talk is, can we find evidence that this part of the brain processes auditory feedback? And I want to turn very briefly to some experiments done in this species, the swamp sparrow. That sings back, in this case, to its own song played through a speaker as a kind of territorial defense. This is called countersinging. And this is just a really handy behavior for very rapidly probing what's going on in the auditory and motor-related domains when a bird switches from listening to singing. These are experiments using a lightweight titanium microdrive to record in HVC during these countersinging bouts. These are little motors that drive extracellular electrodes, and we can use anadromic stimulation to identify different HVC neuron types. So we place stimulated electrodes downstream in RA or X, and then we backfire neurons, and we can use spike collision tests to tell what cell types we're recording from. Okay. And so when we do this in swamp sparrows, and we record responses to playback, and lo and behold, in the awake state, we get these really, really nice responses in the striatal projecting cells, but not in the motor projecting cells. And you can see from this rasterogram that the spike timing is really, really precise. So we're about 8 synapses from the auditory periphery, and we get this beautiful time-logged firing. And when we record as the animal transits from listening to singing, we can see that individual striatal projecting cells are active when the bird is listening, just like I showed you, and they're active again when the bird is singing. And again, the design of this, you might think birds are stupid, but you might recall the first time you heard your voice on tape or on a recording and thought, boy, that's not me. Well, these birds listen to themselves sing and they get defensive and they sing back. But they're actually hearing and producing exactly the same notes and syllables. And these are very simple songs. These are just a monosyllabic trill, so it produces the same syllable, this polynote syllable 10 or 15 times in a row very, very quickly. So we can do these experiments really fast, which is important because the birds are really territorial. And so when they hear the song played to a speaker, they just don't sing, but they fly at the speaker and they've got this $5,000 microdrive on their head. And so they're experimenting often with the last right now. Okay, so you do this and you line up the activity during listening and singing, and you see it's beautifully aligned in time, which we thought, oh great, this is a feedback signal. It's a little bit ramped up during singing. The multi-unit activity in the background is higher, and spikes are in bursts instead of singlets during the listening state, but the same cell's active at the same time. We thought this is a feedback signal, and we went to test this in a number of ways, and I'll show you actually similar experiments we did in the related species of society finch using delayed auditory feedback. The bird sings, we use online syllable detection, and we jam this feedback mechanism by delayed playback of a couple of notes around the time the unit spikes. And if you do this for hours to days, the bird song changes, telling you that you've accessed the error correction mechanism in the bird's brain. And we can ask whether or not this perturbs these striatal projecting units. And the answer is no, it doesn't. So if you look in the singing state, you look at the distribution of spikes in singing, and you look at the listening state, they are aligned, just like I showed you. This is an anchor note in the bird's song. And then you do DAF experiments, and the distribution of spikes is exactly the same. And if you do the equivalent experiment just in the listening state, you play back this distorted song, the unit activity is completely silenced. So the filters on this part of the brain are really, really tight. When you put this kind of distortion in there, the units shut off. So that's a positive control telling us that if this is a feedback sensor, it's not sufficient to change activity. Now I just want to tell you about some new experiments we've gotten really, we wanted to make sure that there was no feedback in this part of the brain, if it's real-time feedback. And we thought, well, maybe it's embedded in the synaptic activity and we can't see it because it's not manifesting change in spike output. So we've been using this little drive device. There's a motor. This is an axon 2D preamp that we reverse engineered. It weighs about 400 milligrams. Don't tell Axon about it. I don't think they're happy we did this. But now we have an intracellular device we can put on the bird's head. Here it is on the bird's head. And we can go hunting for cells. And this is really fantastic work that a postdoc in my lab, Kosuke Hamaguchi, is doing. I'll show you a movie of this. This is, after years of doing intracellular reporting, I thought, well, what's left to do? But I also thought this wouldn't be practical. And it turns out this is uncannily stable when you get it working. And in fact, I think more stable than head fixed recordings, even in an anesthetized animal can be, because the animal can't exert any torque against the electrode. It can rotate its head freely, and the mass of this, the moment arm of this, it only weighs about a gram, so it really doesn't vibrate very much. So I can show you this. You get an idea.
This sharp micro?
Yeah, these sharp microelectrodes. These are the, again, membrane potential recordings, and this is an early movie, so we just splice it into sound when he sings. This is the oscillogram here. There are little cage noises. Okay, and this is also the acoustic, the sound pressure level. So again, these are action potentials. The striatal projecting cells kicking away. The birds eating seeds. This is one of the unnerving things about these experiments is they don't always sing. They spend this time scratching their heads and doing bird things. They're waiting for them to say, yeah. They like to stretch a lot. Wiping his beak, which you can imagine. Scratching again. Where the electrode just stays in there. We've had recordings last up to 6 hours. Most last about 15 minutes. But you get the idea. So now you can get this information out of the bird's head. You can start to, in your mind's eye, he's singing the same song phrase over and over again, and you're starting to get a sense for how stereotyped the pattern of activity is. Okay. And you can then go in, and you can do things. One of the nice things is that you can inject a lot of current into the cell, and you can keep it from spiking. So now you can just measure synaptic activity. And these are experiments like this, where the bird is singing over and over again. We've just shown 3 iterations while the bird is singing normally, and then 3 iterations where he's singing with with delayed auditory feedback. So these are hits and catch trials in red and black, respectively. And you can just see here in these data, these are with confidence intervals, 95% confidence intervals on the waveform. There's just no difference. It's rock solid. And it's really, really stereotyped too. I mean, amazingly stereotyped from one rendition to the next. So we know that the system can be driven by auditory information. I've shown you recordings showing that these neurons can be activated by auditory input. They get auditory input, but in the singing state, the system's totally gated to a motor function. Okay, so in the very last minute, that doesn't answer the question, which is whether or not this system sees effects of auditory feedback. So it could be, for example, that there's no real-time input to the nucleus, but the error correction mechanism still talks to HPC. And to get at this, again, turning to two photon techniques, which have stability long enough to track over many hours reliably, or even days. Katie Cheetah, a former grad student, imaged different HPC neuron types. And she actually used different colored retrograde tracers to distinguish the RA projecting cells from the striatal projecting cells. And the design of the experiment is to go in birds that are late stages of motor learning, make measurements of spine size, in this case, before deafening animals, and then make post-deafening measurements at different times. And the thing about this manipulation is that at this age, the animals are still using auditory feedback information to maintain a song that's almost perfect copy of the tutor song. But over 2 to 3 days, removal of auditory feedback causes the song to degrade. And we can use very sensitive measures to pick up when that degradation starts. And so if we do this and we keep track of who's who, it turns out that there are changes. So now we take the time of behavioral degradation, which is time 0 here, so deafening on average is 2 to 3 days before it's shown here. We align these spine turnover measurements to the onset of behavioral change. We see that there's a selective diminution in the spines on these striatal projecting cells, whereas the motor projecting cells are totally stable. So that tells us that, at least to the best of our ability, we can actually detect structural changes, and I won't show you the data, but we've done whole cell recordings to show that in many analysis that the EPSC input also is diminished in a way that's consistent with these structural changes, that this population of cells somehow gets privileged information about the consequences of deafening, and that those consequences are manifested before the behavior change. It's actually quite a day or more before we detect changes in behavioral output. So it's kind of, in a way, a paradox. The system doesn't acutely sense feedback. It doesn't sense it in real time, not least of these neurons, as best as we can measure. But it does see the consequences of feedback perturbation early enough to actually anticipate changes in behavior. And that gets me to another important point, which is that the changes in spines are actually predictive of the amount of behavioral degradation in a forward manner of about 24 hours. So if we measure spine size index and measure changes in baseline value, if we look at different days, for example, if we look forward one day, if we make a spine size index measurement, and we say, okay, how much is the bird's song going to change? Where more changes down is a smaller percentage here. If the spines are getting bigger, there's very little change. But if the spines are shrinking, there's more change. And if you make this kind of measurement over different time windows, it turns out that the only significant peak is in the forward direction. So about a day. So the structural changes we're seeing in HPC are actually predictive of behavioral changes that will happen on the following day. Okay, so that's the end of the story. What I just told you about is that when a bird hears a tutor for the first time, this motor structure is immediately transformed. The synapses on these neurons are strengthened, stabilized. There's a physiological correlate of this. It's consistent with strengthening of an auditory input into the nucleus. And we can actually manipulate activity in HPC in a variety of ways, showing that HPC is actually participating in encoding that memory somehow in real time, or as close to real time as we can measure. And when we use feedback perturbations, we can also see changes in the structure. They're slower, but they occur in a way that actually anticipates behavioral change. So now we have a part of the brain that's intimately involved in producing behavior, but also gets information both about model and about performance that can be used to guide behavioral change. Now, that's the short take on it. But as I alluded to at the beginning, this is just looking in through a tiny window into the brain, and this is a really distributed network. So you could, if you just took what I said at face value, you could say, well, HPC does it all. Why do we have anything else? But it doesn't. It must somehow interact with the rest of this very big network to achieve this. But what I'm trying to stress is that this part of the brain sees the kinds of information that are necessary to learning, and also controls the behavior itself. All right, so just to, I came to the lab the other day, which is Halloween. My lab likes to dress up on Halloween. This is Todd Roberts and Katie Chivak. She doesn't happen, she's a postdoc now in another lab, she just happened back to the lab, she didn't have a costume, but they did the 2 photon imaging experiments I mentioned. Kosuke Hamaguchi is the postdoc in my lab he's a really good ballroom dancer. And, he's a really good ballroom dancer. I haven't seen him, but apparently he's very good. And he did the intracellular recordings in the same birds that I described. And Malou Mergen, who's a grad student in the lab, developed the optogenetics methods that we used and got them working in bird. And their collaborators is funding, of course, from the National Institutes of Health and NSF, which is both been very generous in supporting this research, and then some really good collaborators, including Michael Brainerd and Michael Fee, Mike Ehlers. I collaborated with other people besides Mike, but there's some real on-backs. And then, just this is our lab. This is a couple weeks ago on retreat in Western North Carolina. Looking at the beautiful mountains. Okay, thanks.
Those are really amazing experiments. I certainly have a question on the technique of the intracellular in the wake animals. So you load an electrode and you have a stepping motor in that little thing that steps you down to the cell?
Show you how this works. Yes, there's a little motor. It weighs about 100 milligrams. It's about the diameter of a pencil lead, and it costs a lot of money. About $800 now. It's made by a Swiss company, Bauhaber. And this is the motor. Here's the motor unit. There's a gearbox that gives about a 50-to-1 gear ratio that converts the torque, generates just enough torque to push an electrode into the brain. And that's connected by a shuttle to this glass pipette here. And then on the flip side is the preamp circuitry. So the whole thing weighs about 1.25 grams, let's say. And this is something, so these are very hard to do, obviously, but it's something, and Kosuke spent a lot of time with this, and we got some really useful help from Michael Fuse Group and Michael Long, who's now at NYU, but the bird's dura is tough, but it's not quite as tough as a mouse dura. And it's kind of built like plywood. There's striated fibers that are running across structure to each other. And if you kind of finesse the electrode tip carefully, you can thread it through the dura of your patient. You break electrodes fairly frequently, but if you start out with 150 to 200 meg electrode, you can get it into the brain at about 100 megs. And that is just kind of the luck of the draw. Sometimes it seems to be very, very stable for a long time. And you can get cells, and I'm sure you've seen this phenomenon where you get kind of a cementing between the tip. It's kind of like a sharp electrode patch. I don't know the mechanics of it, but I know the consequences of it in vivo recordings I've done is you can actually remove the cell body on the tip of the electrode. It sticks so tightly. And when that happens, you can be in the cell for many hours. Yes?
Really beautiful talk. When we go back to the first third of the talk, we talked about the spine turnover. I was wondering about just the control of hearing either another bird that's not the tutor sing or some other sound. In other words, is there this kind of spine turnover change that would just be due to hearing?
Well, they're hearing a lot of things. They're not hearing a tutor song. So they're housed with females. through an early stage of the development until they can become self-sufficient for feeding, which is usually about 30 days. Up till that time, the females are vocalizing, they produce calls, they don't sing though. And the bird can hear its own song. So usually at the start of these experiments, the bird's already begun to produce this isolate type song. So that range of experience is normal, but they don't have the social experience of the tutor, and that would be one potential compound. So we did these experiments two ways. We isolated and then introduced a live tutor. And in some experiments, we raised the birds as isolates and then put them in an operant setting where they hack a key to play back a wave file of the tutor song. And again, in the high turnover birds, that's sufficient to trigger the stabilization. And I may have this slide here that I can show you that that actually, this shows this. This is one of these accidental experiments. So typically we could image for a few days, but once in a blue moon, literally, we got a bird we were able to image all the way from the beginning of the experiment out to the end of sensory motor learning. And this was kind of unusual. This often the tutor just starts singing as soon as the pupil and the tutor are introduced, but this tutor actually didn't sing for a few days. And it only began to sing on this third day, and that's when we started to get a rapid reduction in turnover. So this experiment suggests just socializing with the tutor isn't sufficient, and the often experiments would suggest that auditory stimulation of the song is sufficient. So I think that you have to take these with a grain of salt, because it's not like we do these experiments hundreds of times. These are like collecting hen's teeth, but those sorts of things argue for something special about the auditory experience of the tutor.
Okay, and just one observation I wonder if you could comment. That intracellular recording you showed, it looked like this HBCX projecting neuron is bursting very strongly right at the offsets of the sample. Is that a typical feature?
Not always offset, but Kosuke's analyzing our data set now. He's been able to do something quite remarkable, I think. He's been able to record from up to 100 neurons in a single bird, because he can go back and record over many days or weeks. And he can record from the same cell type. And he finds that these striatal protected cells have prominent synaptic events across the population, at least the population that we can collect, which is on the order of 15 cells of the type per bird, that are most pronounced at onsets and offsets of syllables. And where they occur in the middle of syllables, they seem to be pronounced at acoustic transitions within the syllable. So another way of saying it is that there's sparse synaptic activity that occurs with acoustic change. And that's distinct from a model that's prevalent in this field, that there's a chain-like behavior where there's continuous, we would expect to see continuous synaptic input on the cell. We don't see evidence for that. Not in the striatal projections.
With the isolates at day 60, where you have this enormous variability in turnover, do you have any speculation why there is such enormous variability, and is there perhaps some more subtle timing associated when you do your imaging in the time of the day? So, for example, in terms of circadian rhythms.
Well, we try to be very, very consistent about when we did the imaging. So at least within that level of control, I don't think we could account for it by early versus late night period. But what that tells us is there must be some other factor that contributes to transition to low turnover. And that's independent of experience of the tutor song. So these birds do become committed to a low turnover neuropill and ultimately to an isolate song. And in fact, we think it's probably the stabilization of that work that precludes learning from this experience. And a leading candidate would be steroid hormones. And so the question is why birds that are age-matched have different steroid kinetics. But there's actually very good evidence that if you measure testosterone levels in the yolk of eggs, that it's inversely correlated with the laying order in this species. And that's something, there's speculation as to why that is, but the female invests testosterone in the yolk. And if you're, in this species, they lay 1 egg a day, They lay typically 4 to 6 eggs. So the last egg actually is higher testosterone titer. And the just so story is that helps the birds actually develop more rapidly and compensate for being late in the bird queue. And so they can be more aggressive and kind of, fight for food and stuff and not be disadvantaged by being smaller. But it could be that we didn't assay for testosterone, but that would be a leading candidate for explaining why some birds roll off into this stable state and can't learn. And just, to take it back to the human literature, there's very good evidence from studying language fluency in immigrant populations that the roll-off in fluency, including, syntax, accent, everything, grammar, drops right between about 12 and 14 years of age. So it's arguably something that, the brain, it could be that, at that age you're not distracted by other things, but it could also be that, the brain is changing under the influence of sex stories or whatever it is.
I think they failed to learn with sleep deprivation.
Well, that experiment's complicated to interpret. There are some really interesting correlates with sleep. Tchaikovsky's lab has looked at this, and there's actually evening to morning deconsolidation in song structure in birds that are undergoing motor learning. And the amount of deconsolidation correlates with the end of learning. That correlates with the quality of copy. So the more they deconsolidate even in the morning each day during the learning process, the better copy they make as adults. Sleep seems to drive the deconsolidation, but really establishing that deconsolidation is necessary to learning is it's just not established, but the CBS really supports it.
Can you say a little more about the technique of the two-photon imaging?
Yeah, so this was called two-photon or multi-photon effect. And this is normally with fluorophores, conventional fluorescence depends on an excitatory photon of a short wavelength, resulting in a longer wavelength emission. And so using green fluorescent protein, for example, the absorption for excitation band is about 480 nanometers, that's blue light. And the excitation wavelength is longer than green, about 510, 520.